eukaryotic tree of life. They take up a lot of the diversity and a lot of the branches physically on the eukaryotic tree of life, and which is really exciting. And this diversity, to me, is matched with the ecological roles that they fulfill. They, as both primary producers and consumers in the environment, and more so, their interactions with other microbes at the base of the food chain and food web are really important for the ecosystem. A lot of the ways that I like to study them are through genetic and molecular approaches to really access that unrealized diversity. That was a lot of my PhD work off the coast of Southern California and at the Hawaii Ocean Time Series site. And another thing I really like to think about is how we can tie together the molecular and genetic characterization and thinking about those changes in diversity over space and time, and work to parse the trophic strategies that protists contribute and the trophic interactions that are occurring in these food webs. And so that's what really drives me when it comes to studying microbial eukaryotes. Today, I'm going to talk to you about my core research question right now, which is understanding their role at hydrothermal vent ecosystems. And these hydrothermal vents are fascinating environments. This footage is from our cruise that I'll talk about in a little bit, and back in 2019 to the Gorda Ridge. And these warm fluids are emanating up from the subsurface, and it's quickly mixing with the surrounding seawater. Recall I mentioned I was really excited to parse trophic interactions as they change or across transition zones like oxyclines in the past. And so when I see these environments, I think about that. I think about what's happening where these chemosynthetic microorganisms are supporting this food chain, and then there's this transition zone right where diffuse fluid is coming up from the subsurface and interacting and mixing with the surrounding seawater. So I was really excited about that transition zone. And right in the absence of sunlight here, these food webs are really exciting because they're supported by chemosynthetic bacteria and archaea. And so that brought me to my main goal of wanting to understand what the role of microbial eukaryotes are in this environment. Oh, you missed a slide of vent photos. These are more images from another cruise that I'll talk about today. What do we know about protists in the deep sea so far? There's been a lot of work. The foundational work was primarily microscopy and culture-based studies to really see that protists are barotolerant and able to survive in these extreme environments. Then, of course, with sequencing approaches, we were able to access more of the diversity and understand that the protistan community might be quite diverse at these deep sea ecosystems. So while there's been a lot of work on that, there's not as much on microbial eukaryotes as there are on the bacteria and archaea that inhabit these sites. So that's another thing that drew me to studying deep sea vents, of course. The other thing, in addition to characterizing the diversity and the presence of protists at these hotspots of primary production, one of the things that the sequencing definitely and some of the culture-based work revealed was that there's species that are similar to heterotrophic organisms on land and other places that we study them more easily. And so their role as phagotrophs, or consuming smaller prey, microbes, bacteria, archaea, and smaller eukaryotes, was definitely hypothesized and was even observed. In these early accounts, these images actually have little vacuoles with bacteria inside of them, and some organisms were brought shipboard or in labs from the deep sea and fed bacteria. In one case here, this is from Guaymas Basin, where a ciliate was isolated, brought shipboard, and fed this analog prey, which is glowing green in this image here under epifluorescence. And you can see that the ciliate has in fact consumed prey. So we know that this trophic interaction is occurring. And so I wanted to hone in on this even more and work on quantifying this interaction. So like I've kind of highlighted this main arrow here, the consumption of microbial prey. Today I'm going to talk about grazing incubations and sequencing approaches that I use to address this question. At first, to give you a spatial idea of what I'm doing and recall that transition zone from the background to the vent fluid that I'm really excited about is making sure I sample when I'm at these vent sites, the background plume, and then the diffuse flow. And for the diffuse flow via ROV, of course, we're sampling fluids that are less than 100 degrees C. Or that's what I'm targeting. And so I'm taking vent fluid and background and plume fluid and filtering this in situ, hopefully for sequencing. And today I'm going to share sequencing results that are from an 18S tag sequence survey. So it's a little bit it's a barcode approach where I take that extracted genetic material, isolate that conserved gene, amplify it and sequence it, and then I'm able to assign a species level designation, which I'm going to reference as an amplicon sequence variant. And it's kind of like a barcode approach. Clearly, I did my PhD in California, remnants of that in these talks. So that's how I'm going to approach the characterization of the community, because this gives me a snapshot of who is present in each of these sites. And then the other thing I'm going to talk about are the grazing incubations. So from the same locations that I'm getting this vent fluid, I'm actually performing grazing incubations, which I'll talk about. But I'm also connecting this to some of the sequencing material, because while these grazing incubations address what is the grazer population doing in this environment, I'm also taking material after the incubation and sequencing it. So I want to continue on talking about the grazing incubations in a little bit more detail. And the first thing, of course, is actually collecting the fluid, like I mentioned, with the help of the ROV. In this video here, this is actually from a vent called Vente Latte from the Gorda Ridge. And this has a temperature probe at the end, again, targeting that low temperature diffuse vent fluid. This fluid is collected, brought shipboard, and then I'm spiking this with a fluorescently labeled prey. And so this prey, or FLP, is that analog prey, kind of like I mentioned in the Guaymas Basin study. And here is an image of that FLP in glowing green here. And this is actually from some of these experiments where I'm showing the size of the in-situ bacteria from the hydrothermal vent ecosystem, showing that it's a prey analog for what the resident prey community looks like. And this is an isolate from the Huber lab, Hydrogena vibrio. So I have my vent fluid shipboard, spiked it with FLP, and now I work on incubating this. And so I mentioned this because there's two ways to approach this, and I have experiments from both. The first one is I'm going to talk about is an FLP disappearance approach. So this means that FLP is in my incubations, and then I'm counting the labeled prey as a tracer as it disappears from the experiment or from the incubation bottle. And then from this, I have my time zero and time final, and I can calculate or estimate a grazing rate from the site. And the other approach is an FLP uptake. Similar idea. From the time points that I take, instead of just tracking the labeled prey decreasing over time, I'm actually estimating and through the microscope, counting the number of FLP I see inside of the eukaryotic cell. So then I have on the y-axis instead the FLP per eukaryotic cell over time. And then the slope of this line helps me calculate that grazing rate. And I go into a little bit of detail. These are pretty laborious to run. I want to mention that I do this in replication as much as possible. And I'm running a no grazer control to make sure that the FLP isn't disappearing for another reason. So the first study site is Gorda Ridge. And this was Gorda Ridge is off the coast of Southern Oregon. Axial seamount is to the north here. This is a tectonic spreading center. And we were able to visit this site in 2019 with a subsea, under the subsea program partnership with NASA NOAA. This was via EV Nautilus with ROV Hercules, which I'm sure you guys are familiar with. And this was with Ocean Exploration Trust. This was really fun to be on. And so the Gorda Ridge right here, this is where the Pacific plate is separating from the Gorda plate, and that's creating that spreading center. So here, these are the 4 main vent sites that I focused my studies on for this expedition. Mount Edwards, Vente Latte, like I mentioned before, Candelabra, and Servensalot. And just a reminder, targeting a low temperature diffuse flow, and in this case, looking at the FLP disappearance experiments. This is what the product of that looks like. So here on the y-axis, I'm showing the FLP cells per mil. So that's that labeled prey that I'm tracking the disappearance of over time. And in the diamond symbol is the near vent background fluids, or my background representative experiment. And these 4 lines represent the 4 vent sites. So over time, we're seeing consistent FLP loss. And this is representing to me that there's a predation pressure happening at these vents, which is really exciting to see. Then I can actually use the T0 and T final values to then calculate that grazing rate. And so here's the same symbol and colors represented with the background, and then my 4 vent sites where I have the cells consumed per mil per hour along the y-axis. And I was really excited about these values because we're getting a grazing rate that's higher at these vent sites compared to the background fluid. So when I think about that transition zone again, from going from background to the diffuse flow, we're seeing an uptick in that activity, that grazing activity, which is really neat. Another thing that we did at Gorda Ridge, of course, was to get DAPI counts from the prokaryote community. So I have a biomass value, or a representative biomass value of the prokaryote community in situ that's potential prey. So then I can also estimate of the prokaryote biomass in that vent system, what percentage of that might be taken up by protists via grazing per day. And that's what these prokaryote turnover percentage values represent. So as an example, Servensalot on the far right here in this pink color is showing one of my higher grazing rates estimated for this system, over 1,800 cells consumed per mil per hour. And when we consider the prokaryote biomass at Servensalot, we're seeing a prokaryote turnover percentage of 62%, and on average. And this is average across my replicate experiments. And the way that I like to interpret this is that 62% shows to me that there's a higher, the system, the microbial food web is under more top-down pressure at Servensalot compared to sites where that prokaryote turnover percentage is much lower, like the background. Okay, so now we're starting to be able to see that there's that trend in the background versus vent site. Okay, so I'm gonna come back to these 2 questions of why we, I hope that I've outlined a little bit at least of why we care about this, and I'll come back to addressing who's responsible, who are the grazers that might be responsible for this activity. But now I want to actually shift over to the other study site and look at the grazing rates that I was able to get from that. And that's from Mid-Cayman Rise. So this was a Mid-Cayman Rise, this was a cruise last year in January 2020, where we were on the Atlantis with ROV Jason. And Mid-Cayman Rise is a really neat site. This is an ultra slow spreading center in the Caribbean, home to one of the deepest known vents, Picard. And there's actually two main venting sites within the Mid-Cayman Rise, and they're physically relatively close to each other, but they're also geologically distinct. One of them is Von Dam at 2,300, about 2,300 meters, and Picard, which is closer to 5,000 meters, and so that's the deepest one, of course. And a quick note that Gorda Ridge is closer in depth to the Von Dam site here. So this is what some of the vent ecosystems look like. there's a very strong shrimp theme essentially here is the main take home from those images. But it looks a little different than Gorder Ridge site. So in addition to running those grazing experiments shipboard, similar to the way that I collected that fluid and set up my experiments at Gorder Ridge, I was also able to run some in situ pressure. So this was with a collaboration with Jeff Seawald, where I was able to collect vent fluid via these isobaric gas tight chambers. What happens is the IGT samples the vent at depth here, and it remains in this chamber at the in situ pressure that it was collected. And then shipboard, I maintain that pressure, introduce the FLP, and then run my experiment. And for all the experiments run on this expedition were the FLP uptake. So they're shorter experiments actually, and I'm working to track the FLP per eukaryotic cell. So to reiterate that a little bit, I do have two images here that show what that process looks like, and I'll get back to why you should care about this. But here is two eukaryotic cells imaged from some of my experiments, and you can actually see the DTAF stained FLP inside the cell. So this is me counting 2 FLP per euc cell and then 1. So let's look at some of these results. On the y-axis, I have the cells consumed per mil per hour, same units as what I previously showed. And then on the far left, I have the background and plume sites. So that's my background environment. And then these are all of the vent sites from mid-Cayman Rise. And the Picard samples are in a red symbol, and then Von Dam is represented by this blue-green color. And I also differentiated which samples were run shipboard by bag in the circle symbols, and then the triangle symbols represent which grazing rates were obtained via the IGT approach at in situ pressure. So a couple things to note. One, I have these super low 0 grazing rates, and this is because these FLP uptake experiments sometimes have a 0 or negative slope. And so I've kept them in here for now, but this means that the grazing rate was not detected at these sites, or below detection perhaps. The other thing, regardless of those 3 experiments, I wanted to point out that this is my background and plume grazing rate is lower compared to most of my results from the venting sites. So this is really exciting because this is starting to show us that these grazing rates that I'm getting at another hydrothermal vent site, I'm seeing that same trend that I was so excited about personally, where I'm seeing an uptick in protistan activity and specifically grazing at the site of venting fluid. So the other thing to address is the shipboard versus the IGT. So I ran 9 total shipboard experiments, and I think it ended up being more like 7 or 8 total IGTs, and some of them did not end up working. But what's interesting here is that the IGT and shipboard experiments, for the most part at the vent sites, are comparable to one another. The ones that gave me low detection, of course, were from shipboard, so it's something I'm still working on processing some of this preliminary data. But to me, I'm not going to throw out my shipboard experiments. Is one of my take-home messages. I think that they're still offering a lot of information with respect to constraining these important values in this food web. And so the grazing rates from both shipboard and in situ were comparable to one another. But of course, as we know, running things at in situ conditions, no matter what, is always a best practice. So I will talk briefly later about ongoing efforts and future efforts to continue running things at in situ. But I was quite encouraged to see that the end result was comparable to one another. So the other thing that I'm excited to share is that I was also able to compare these results with cell concentration and biomass with both the prokaryote community and the eukaryotic community. So this is the same type of figure, background and plume on the left-hand side on the x-axis, and all of my vent sites, same symbols and color schematic. But now on the left, I'm showing the prokaryote cell concentration in cells per mil along the y-axis. This is derived from DAPI counts using similarly collected vent fluid. And on the right, I have the eukaryote cell concentration. So because I'm doing these FLP uptake experiments, I'm counting the euc cells as I go along, essentially. And so I'm actually able to get these values. And which this is really exciting to me. I don't have any other examples of eukaryote cells per mil at deep sea hydrothermal vent sites. But if someone else does, please let me know. I'd love to compare notes. So, and the main take home from this is that we're seeing there's an elevated concentration of microbial community concentration, population abundances at the site of venting fluid. And this makes sense. This has been hypothesized before and seen in many instances. These values are consistent with other work at deep sea vent sites, but it's exciting to see the same trend with the protist community. Now, recall I'm doing these FLP uptake experiments. So in addition to searching for euc cells in my preserved fluids when I'm doing microscopy, I'm also able to image as many of them as I can and then measure them. And with that, I can use field standards to estimate biovolume and get actual carbon per liter estimates, or carbon per cell. Use carbon per cell field standards and estimate the micrograms of carbon per liter. For prokaryotes, in this case, I've used a reference with 86 femtograms of carbon per cell and convert this to biomass. And then for eukaryotes, I've done this based on the measured cells. And so this is, of course, a factor of the eukaryotic cells per mil, but I will show a figure in a moment that relates of why I'm so excited to get these values. So to reiterate that point of seeing this really neat trend of increased cell concentration for both prokaryotes and eukaryotes, and then bring this back to the grazing rates. This is now all the grazing rates from both sites. We have Picard and Van Dam in red and blue respectively, and then Gorder Ridge in the green color has been added now. On the left, I have the background results from both sites in cells consumed per mil per hour, and then all of the results from the hydrothermal vent. So we're seeing that this trend is comparable when you look at the background to the vent sites at two separate hydrothermal vent ecosystems. And this is consistent, of course, with the increased microbial concentration seen and biomass at the vent sites. So let's now finally talk about why do we care about this in a little more detail, and trying to bring this back to the food web. So this is right, I'm thinking about my transition zone and looking at just the values from the vents for now. And previous work from Jesse McNichol, actually using the IGT experiments from East Pacific Rise, he ran a series of incubations in IGTs using substrates to estimate the primary production being produced from the chemosynthetic population at crab spa at EPR. And this is the range in micrograms of carbon per liter per day that was estimated. So we have this, to me, it's like starting out value of what's being presented, what can be produced by the population. And now we can also, I've taken those grazing rates that I just showed from Gorder Ridge and Mid-Cayman Rise, and now changed them to, or converted them to show the amount of carbon that can be consumed by the protistan in the same units of micrograms per liter per day. So we see there's a little slightly different range, but they are comparable to one another between the two sites. But now we're starting to see that the protistan community can take up maybe potentially a substantial portion of that carbon being produced in this ecosystem. And this is exciting because then we can think about the microbial loop and think about how carbon is not only being taken, after being taken up, also being exported and recycled in this environment. Because as we know, protists play an important role as primary consumers in a lot of environments, and that contributes to carbon export and nutrient recycling in the ecosystem. And so I'm excited to think about these values and assign quantitative measurements and really work to constrain this carbon budget. The other thing, oh yeah, that's right, one more number. So from the estimated biomass of the protists, I also can estimate their carbon pool. And this is my carbon conversion for eukaryotes based on what I measured. I think it's potentially a little bit low, so perhaps these are a little more conservative estimates. But by looking at this pool of the protists, I can view them now with a carbon currency context, essentially. And I also think about them not only as consuming prey and placing predation pressure on the chemosynthetic population, but they themselves are prey for other heterotrophic levels, other protists that might be larger than them, for instance. And so getting these values is again, really exciting. I mentioned these two papers as well, because another thing that kind of hits to the broader implications of this is that we think about carbon export throughout the water column, and some of that is reaching the deep sea, of course, and depending on where you are in the ocean. But some of these papers talk about the organic carbon exported locally to the vent site, what's happening right around the vent. Particularly POC maybe coming from the plume environment and then being recycled in the vicinity of the hydrothermal vent ecosystem. And so that potentially might outweigh what's reaching that deep sea area from export from the euphotic zone, for instance. So this is one of the ways I'm excited to kind of continue tackling this question and thinking about the kind of like the mosaic of deep sea carbon routes, right? Okay, so now I'm going to shift gears again and go back to talking about who are these grazers. And to remind you, running these concurrent grazing incubations and also collecting samples for in situ, I'm going to present some of the sequencing results. And I'm going to remind you of my hypothesis that I've been hinting at and mentioning a lot, where this community is likely enriched at these hydrothermal vent sites. Now, this is backed by increased grazing rates and increased cell concentration and biomass. So this is the results from the sequencing survey from Gorda Ridge. The results from Mid-Cayman Rise are still being processed right now. But on the y-axis here, I have the relative sequence abundance. And then on the x-axis is all of the Gorda Ridge samples, where I have all the background samples here. And then this is in situ from the plume and vent sites that I was able to get material from. So the colors designate the different protist taxonomic groups that I'm showing here. The main thing, there's two main things I want to point out. One is that the background environment looks a little bit different from the vent and plume environment. And this makes sense. Most of the taxonomic groups are still represented in both cases. And I'll get into a little more details about this in a second. But many of the main taxonomic groups are found in different relative abundances, particularly the ciliates in this light pink color, Cyndineales, and then also some of the radiolaria. The other thing I want to point out is this isn't all of my data. I also have results from sequencing from the grazing incubations themselves. And that's, now I've filled this in, and in addition to the in situ and in bar plots, I have now added in the grazing. And if you draw your attention to maybe just one of them here, the Mount Edwards site, for instance, we have the in situ and then the 2 grazing experiment results. And one of the reasons I did this was I wanted to compare what the in situ community looks like, and then when it's brought shipboard, to see if during my grazing assays, am I honing in on the heterotrophic community? How much is that bringing shipboard modifying the community? And to me, this is telling me that there's a little bit of modification, of course. I'm bringing fluid shipboard, but the community is still very much representative of the in-situ. The community in the grazer, the end of the time final of the grazer incubation is still very much representative of the in-situ community. So this is encouraging to me. So the other thing I wanted to go over was talking about how else I can kind of slice this sequence data. And one of the things I wanted to address was what might be enriched. And so I separated the sequence data based on presence throughout the hydrothermal vent ecosystem, meaning cosmopolitan, compared to sequence data that was found only at the vent sites themselves and not present in the background or plume environments. And the results are split with respect to total number of sequences. But when I look at the total number of ASVs, meaning that species level designation of my genetic signatures, I see 500 and change of the cosmopolitan community and many, many more of the resident. And to me, this indicates that I have higher species richness, just localized to the vent site, much higher total number of species, not necessarily total number of sequences though. So I'm gonna talk about the differences between the cosmopolitan and resident, but I'm just gonna talk about the ciliates for now because they were a pretty prominent component of the community in Egg-Order Ridge. So here I have the ciliate community separated by family and class listed here. And then this is the same x-axis as the bar plot that I just showed. And I've separated the ciliate community based on membership to that cosmopolitan, so found throughout, versus the resident. And the first thing I want to point out is that I see a consistent signature within the ciliate community, meaning these bubble plots show the total number of ASVs. And we see that there's, again, a higher total number of ASVs among the resident ciliates. And now when I actually fill in the heat map showing the relative abundances of this data, we see again there's more total resident ciliates than compared to the cosmopolitan. And this heat map is actually a centered log ratio data. So I took my sequence data and transformed it. So the white represents the geometric mean of all the data shown here. And then the pink colors show where the sequence abundance is higher than the geometric mean, and the green is the inverse of that. So the couple things I want to point out, I've reiterated the species richness component. The other thing that's neat to draw from this type of data is that right away I can see there's certain groups that are completely absent from the cosmopolitan. Some of these might be involved in symbiotic interactions as well, which is another exciting component to look into in the future. But this indicates that these are classes that are potentially endemic to the vent site. So to me, they're good candidates to go back and try to get more physiology data from, or metabolic potential information from, to see what traits are found among the ciliate endemic community. And then the other thing I want to point out with these blue arrows now is that these are groups that are found in both the cosmopolitan and resident, but they're comparatively, they're more enriched. There's higher total numbers of them, more pink squares on the resident side, so they're more represented in the community at the site of the venting fluid. And so when I talk about honing in on who those heterotrophic members of the community are, these are some of the big players that I think of right away. That's how I'm working to connect this data back to the grazing rates that I saw. So this was in summary, all right, I'm showing that there's predation pressure that's greater, likely in response to prey availability at the site of hydrothermal venting, and that also brings with it higher concentrations of protists, higher biomass of protists at these environments. And we're really working on getting those values that constrain food web models, for instance, at these hydrothermal vent environments. And it's really exciting to get quantitative measurements of the impact of that protistan grazing. And I think there's a lot of implications for deep sea carbon cycling. And I mentioned some of them in my food web map previously, but it's an exciting area to continue exploring for me. The other thing I want to mention is the in-situ experiments. So we saw that perhaps the shipboard and in-situ are comparable to one another, but future work is definitely going to continue tackling this question. So we have a funded proposal to go back to axial seamount, and this obviously this is not going to happen. This is going to be a later year, TBD, essentially. And so at axial seamount, we will be deploying this MS-SID device that can run these grazing experiments in situ. And so it'll be put down next to the vent site, collected vent fluid, and actually run those experiments in situ. And Maria in biology here at WHOI has run these before. And I'm continuing to explore the genetic signatures of these organisms at this site. And I have archive samples from previous work that Julie's done. I'm working on processing those, especially because this is TBD. And another thing also TBD, including the date now, is work through SOI. So we'll be joining their ship in the future, and also deploying this MS-SID device to again emphasize the being able to run these experiments in situ. And then finally, I just wanted to remind you that I also looked at that in the enriched community at the site of the hydrothermal vent fluid, looking at putative endemic taxa, and I think there's a lot of really exciting underexplored metabolisms yet to be explored. And so I'm doing a lot of meta-omics as well to kind of follow up on some of those questions. And I want to list my collaborators and acknowledge all the people in my lab. Many of these people are also co-authors on this paper, from Gorder Ridge, captain and crew, and people and collaborators that make all of that work possible. And with that, I will take questions. Thank you.
Great, thank you, Sarah. That was an absolutely fantastic presentation. If anyone has questions, feel free to unmute yourself, or you can throw them in the chat as well if you don't want to unmute.
Hi, Sarah. Thanks for a great talk.
Thank you.
I was struck by, I guess when you first presented the different areas that you were sampling, I would guess there would have been a gradient from like the background to the vent, but it looked like the plume was closer to the background. Did that surprise you? Or
the plume was closer to the background for grazing or for the molecular?
For grazing and like abundance and yeah, those metrics you were looking at.
Yeah, I'm still working to parse out what the plume signatures mean. They are more similar to background. I wonder if it's just like the physics of the plume are more similar to the background environment, and so I'm seeing like a more dispersed community. So then that looks more like the background environment. One of my hypotheses going in was that the plume was going to look more like the vent. You're right, but yeah, that was really striking.
Hey Sarah, it's Roxanne.
Hi.
Thanks so much for a great talk. I have lots of questions, but I will keep it to one. So I was curious about oxygen in your experiments, because a lot of the taxa that you saw enriched are anaerobic ciliates. And so yeah. I would imagine if you have a contained bag or IGT that if there was oxygen to start with, excuse me, it's getting depleted throughout your experiment. So I'm curious if you have any sense of what the oxygen concentrations were in your experiments.
I do not have a sense of that. I can imagine that whatever for the FLP uptake experiments, I definitely could see that was depleted over time. So that could definitely contribute to it, and I'll definitely think about that as I process the time zero or in situ versus the time final with respect to the sequencing data. And the IGTs might be an extreme example of that. The IGTs are not enough fluid to collect the molecular sample, so I don't have the end result of that. But especially because we do find anaerobic protists in these vent sites, and what's happening to them when it comes, because there's still oxygen nearby in the area. And so I don't have a solid answer. I don't have oxygen data from these experiments right now. I think that there's some that I might be able to get from Mid-Cam and Rise, but I know they're still being processed. So the story is evolving.
Thanks so much.
Hello, Sarah. It's Karen Wishner. I guess I'll put my video on there. I was just wondering if anyone is following these up the food web to a little higher trophic levels. I know that there are vent-associated zooplankton and probably some filter feeders in the vicinity. Is anyone looking at whether these protists are a food source for those organisms?
Not that I know of. If someone does know of this, I would love to talk to them. I mean, I think that, like I showed, that I know that there's a carbon pool that's to be eaten, and maybe a substantial amount of prey and food and resources. I know there's people that do study those organisms, and so I'd love to talk to them more about putting all the arrows and boxes together for that food web. Yeah.
Great. Yeah, that'll be real interesting.
Sarah, this is Tatiana.
Hi.
Hi. Nice talk. That was really interesting, and I'm looking forward to talking to you later today. But question I want to ask, because we've got a lot of students in the room, is you're heading into a habitat that, as you pointed out, people really focus on in terms of bacterial metabolism and bacterial activity, and not so much the protists. So I'm curious, as you looked at those sequence data, how much of it looked like organisms we know and love from anaerobic sediments in shallow or deep habitats, aerobic habitats in the surface ocean? Like, did you see any kind of shifts in the composition or even unknown sequences that lead you to think there might be some endemic species in those habitats?
Yeah, I mean, the answer is yes. I did see a lot of different trends and really distinct diversity signatures, in some cases vent to vent. I can actually just show, maybe I can just show this for fun. Oops, too many tabs. I showed just a snippet of this, and this is not presentable because it's so large and kind of hard to read, but from a bird's eye view, right, these are my different taxa, and this is just the breakdown of them. I'm seeing a lot of diversity I'm seeing a lot of differences in some of the population that's primarily endemic. And so, but you're right in that there's a lot of unassigned things. And I don't mean things that come back to me assigned, but things that are assigned down to maybe the supergroup only or the family level, and I don't have any more information. And so I think my approach is, there's kind of two ways that I approach this type of data. One is to work on getting to a place like this where I can at least try to diagnose the trend spatially in this case. So cosmopolitan versus resident, the different vent sites, working to connect those to maybe some environmental parameters. And another approach is to always be kind of thinking about the sequence data. I think a lot about bioinformatics and reproducibility. And so I'm always trying to make sure this data is available, reusable, and I'm continuing to do HNS surveys so I can hopefully link these studies to one another, and hopefully continue building that, even if we end up with a lot of ASVs that aren't assigned or belong to species that we don't have an idea of what they are exactly. We can at least start to get the ecology of where they might be present, or the biogeography of them, so then we can eventually assign that to a species. We have that background context. I'm not sure if I answered your question in full.
Yeah, no, absolutely. And I just wanted to, that answer is really helpful because it really highlights how much room for exploration there still is at those vent sites. We get there so rarely. I mean, really frequently now compared to even 2 decades ago, but relatively rarely. And yet there are important groups of organisms where it looks like there's just no match at any sort of reasonably fine resolution of
Yes.
taxonomy. So to me, that's really interesting. A lot of
Yeah.
explorations that go.
No, I love doing that. When you get your data, I feel like I look at it, I'm like, okay, this is a bunch of unknown stuff. I'm gonna just apply these methods and see if I can make sense of something. And then, you know, it's little pieces of the puzzle that I'm trying to put together. So yeah.
I don't know if you can see the chat, but there is a question there. I can read it if you can't.
I think I can figure it out. I think I figured out Zoom now after all this time. Classification of microeukaryotes as cosmopolitan versus resident. Oh yeah, so the criteria for cosmopolitan versus resident. I use the basic just presence absence of those ASVs. And so if an ASV, meaning that species level designation, was present only in the background, or was present in the background and throughout the vent sites, even if it's just scattered and in the plumes, then I consider that cosmopolitan. But if an ASV was present only in the vent sites and not anywhere in the background, then I classified it as resident. And I like this last part of the question of, do I expect the classification of a cosmopolitan versus resident to change? And I would expect that. I'm excited to get a couple more sequencing runs done from the 18S survey, because I think I can, using again that kind of binary approach of presence absence in the background versus the vent site, and really look at if there's patterns or similarly endemic taxa at different hydrothermal vent ecosystems. So I think it will change a little bit as I get more pieces of that puzzle.
Hi, Sarah. I have a question. This is Ian Bishop. Great talk. Since we're talking a bit about classification and some of this compositional data, that big chart that you just showed, I noticed that there are a bunch of like epipelagic community members, like stuff we would expect from exogenous or allochthonous systems. And I'm just curious, I guess I didn't catch it in your talk, how you're sampling that. And I'd love to hear your comment on how much of that collected sample you expect, given the way you're collecting it, to be from outside the system itself.
So you're talking about some of the like
Fermentopile, various autotrophs. Yeah, exactly.
Yeah, well, yeah, just to pick up on one thing you just mentioned, I'll put this slide back up. I just don't like showing overly complicated things, so I'm happy. There's a preprint of this as well with this figure. Yeah, so just to pick up on one thing you mentioned, the stromatophiles are really, I think, really exciting. One of the organisms that was very prominent in this kind of near background, but still close to the venting sites, was the Cafeteria ACA class, I believe, yeah. And so there's a lot of stromatophiles that are heterotrophic, some of the mast, and those organisms I'm excited to explore more, because there's a lot of work from the Malaspina expedition throughout the mesopelagic showing the importance of a lot of those stromatophiles as heterotrophs throughout the ocean. But yeah, the ones that are I would consider primarily phototrophs, or maybe mixotrophs that are primarily phototrophs. They did show up in my data. I wasn't surprised. I've seen this in every sequence survey from deep sea vent sites to date for protists, but they did not make up a significant portion of my data at all, and so I wasn't that concerned. That's another reason I started down that classification, because I wanted to see if there was if they were primarily cosmopolitan, so maybe around in the background environment and sinking material, coming in via sinking material, I might provide more evidence of that. Yeah, I guess I'm not concerned about them in my data, if that's what you're asking.
Yeah, great, thanks.
Okay, well, if there are no further questions, students and postdocs, feel free to stay on and we will have a meeting with Sarah. And let's just unmute and give her one last round of applause.
Thank you all for coming.
Thanks everybody for staying. We have a big group. Yay. If
Hello,
everyone
thank you
just
for
wants
coming.
to go around and introduce yourself to Sarah, I will start, I guess. You already know me, I'm Victoria, and I study the distribution and effect of plastic pollution in coastal ecosystems. And Heather, you want to go next?
Hi, Sarah.